Okay, in this video, let's take a look at the pentose phosphate pathway. Now, one thing to keep in mind is that the pentose phosphate pathway occurs in all cells. But I'd like to focus this on the liver because of the product of the pentose phosphate pathway. The major product is going to be most important in biosynthetic reactions in the liver. Now, let's begin discussing the pentose phosphate pathway, beginning with the different products that are formed. Now, recall from the energy-producing pathways, we are able to reduce NAD through enzymatic reactions, dehydrogenase reactions, to generate NADH. And recall the purpose of generating NADH is to put this product into the oxidative phosphorylation pathway in order to generate ATP. So the sole purpose of NADH is for energy generation. Now, a slightly modified product is made in the pentose phosphate pathway. In this pathway, we are going to take a related molecule called NADP. NADP is going to be reduced in this pathway to form NADPH. Now, the only difference between NADPH and NAD is simply a phosphate group. But the function of this cofactor is extremely different from NADH. NADH, of course, is involved in energy generation. NADPH is involved in biosynthetic reactions. Now, why do we need NADPH in biosynthetic reactions? Recall that in order to create a larger molecule, we are going to need, this is going to be a positive free energy change, because we are constructing bonds. In order to construct these bonds, we need to put energy into the reaction. We are also, in this reaction, decreasing entropy, because we are creating a higher ordered molecule. All of this combined, creating the bonds, decrease in entropy, is going to require an input of energy, so therefore it is a positive delta G. So the energy for driving this reaction will, of course, be the nucleotide triphosphate molecules such as ATP, GTP, UTP, and CTP, but we are also going to require reducing power. And the reducing power is simply going to be needed for reducing smaller molecules in order for them to be constructing larger molecules. This reducing power is coming from NADPH. So NADPH is required for biosynthetic reactions, NADH is required for energy generation. Now let's take a look overall at the pathway for generating NADPH to see where this comes from. Now recall, we are going to be focusing on the liver, and so in the liver, in the fed state, recall we have insulin signaling in the fed state, we have GLUT2 on the cell surface of liver. Recall that this GLUT2 is going to be working only when glucose concentrations are high in the blood, and this, of course, is during our fed state. So recall the first reaction of glycolysis in this reaction is to generate glucose 6-phosphate. This, of course, by this particular enzyme is called glucokinase. Remember that glucokinase is paired with GLUT2, whereas hexokinase is paired with the other glucose transporters. Now, from this step here, we can use our isomerase reaction, our phosphoglucoisomerase, to generate fructose 6-phosphate as we begin to proceed down the glycolytic pathway. And then the next reaction, of course, is PFK1, phosphofructokinase 1, and this is generating the fructose 1,6 bisphosphate molecule. Once this continues down, we continue down through glycolysis, TCA cycle, oxidative phosphorylation, and the end result is to generate large amounts of ATP, especially in the fed state when we have a lot of resources available. Now, recall back in glycogen synthesis. In order to synthesize glycogen, we need to regulate PFK1, which is our gatekeeper for glycolysis. And that is done by the intra-pathway regulation, which is the levels of ATP. So ATP levels, as they rise, recall, will inhibit the formation of fructose 1,6-bisphosphate by inhibiting PFK1. This will cause fructose 6-phosphate to increase in concentration. When this happens, recall that the isomerase reaction is reversible, and this will shift back into glucose 6-phosphate. Now at this point, glucose 6-phosphate cannot reverse back into glucose because this is not a reversible reaction. This is such a large negative delta G that we are inhibiting the reverse reaction. Now for glycogen synthesis, we shift the glucose 6-phosphate into glucose 1-phosphate by our phosphoglucomutase enzyme. And then recall this is charged by UDP glucose formation. And then from here we can form glycogen to store the glucose. Now there's an alternative path from glucose 6-phosphate. And this path is going to be to the pentose phosphate pathway. Now again, regulation into this pathway is going to be dependent on the inhibition of PFK1 because we have to prevent our substrates moving into glycolysis. So by high ATP levels, the cell is fully charged. We inhibit PFK1. We reverse our fructose 6-phosphate to glucose 6-phosphate reaction. And now we have two paths to go. We can either enter glycogen synthesis or the pentose phosphate pathway. Now, what is going to determine entering the pentose phosphate pathway? Well, the first reaction we'll take a look at in a second is going to be formation of NADPH. So the quantitative levels of NADPH in the cell will determine if glucose 6-phosphate will enter the pentose phosphate pathway. So the very first step is the committed rate-limiting step. So for example, if NADP levels are high, that means that the cell is deprived of reducing power for biosynthetic reactions, and so glucose 6-phosphate will enter the pathway. If NADPH levels are high, so we have plentiful reducing power, that will inhibit the first step and prevent glucose from entering this pathway. Now the other product of this pathway are pentose carbohydrates. There's a couple of pentose carbohydrates that are important for cellular activity and function, but the main one to be considering is ribose 5-phosphate. So recall that this is the backbone carbohydrate structure that is necessary for constructing DNA and RNA. So when we need carbohydrates for our DNA and RNA synthesis, this is the pathway we're going to rely on to generate the 5-carbon ribose structure. Now, with that in mind, let's take a look at the reactions a little closer. So the first step in the pentose phosphate pathway abbreviated PPP, is we are going to take glucose Again, we are going to take glucose. It'll be phosphorylated on the number 6 carbon. Glucose 6-phosphate. And again, as I said before, the first step of this reaction is going to be a redox reaction. And so in this reaction, we are going to be converting NADP to NADPH. The enzyme that does this is called glucose 6-phosphate dehydrogenase. The carbon that we are oxidizing is the number 1 carbon. And so with that, we are forming this structure. Now recall that when we perform a redox reaction on a carbon with an alcohol, we are going to be taking the electrons associated with this hydrogen. The electrons that are associated between the oxygen and hydrogen will collapse down into a double bond between the number 1 carbon and the oxygen. So we are forming, again, a ketone structure. This structure is called 6-phosphoglucono, representing our glucose backbone and the 6-phosphate lactone, because we have a ketone structure. So the most important part of this reaction is that it is the entry level reaction. So this is the entry step into the pentose phosphate pathway. This enzyme, glucose 6-phosphate dehydrogenase, is the regulated enzyme to control entry into this pathway, and it is controlled by NADPH. So high levels of NADPH will inhibit this enzyme. Now, recall dehydrogenase reactions have a fairly large negative delta G. And so that makes this reaction irreversible. So once we pass this point, we are now fully committed into the pentose phosphate pathway, and we cannot return to the glucose 6-phosphate. So the next step of this reaction is a simple one. This enzyme is called lactonase, and this is simply a hydration reaction. We're going to be opening up the 6-membered ring structure, and we're going to add water across the number 1 carbon to form an acid. And what we have formed here is called 6-phosphogluconate. Again, reflecting the 6-carbon molecule, glucose. Phospho, we still have our phosphorylation on our 6-carbon. Gluconate, ATE, represents the acid form. And when the water adds across this bond, of course, we are breaking open the ring structure. Now, the next step in the reaction is going to be another redox reaction. And in this redox reaction, this is a special reaction because we are now reducing ourselves down to 5 carbons. So this is not only a redox reaction to generate NADPH, but it is also a decarboxylation reaction. The enzyme that performs this is called 6-phosphogluconate dehydrogenase. And the structure we are forming from this is a ketose. This ketose is called ribulose 5-phosphate. Ribulose now, because we are a pentose, we still have the phosphorylation, but is now in the number 1 carbon because, number 5 carbon, because the number 1 carbon was ejected by the decarboxylation reaction. Now the final step of this reaction is to generate the ribose 5-phosphate structure. And this is a phospho-pento-isomerase. So we are simply performing an isomerization reaction. And now, instead of a ketose, we are forming an aldose. Now this molecule, of course, cyclizes to form our 5-membered ring structure of ribose. Now, one more note is that the cell is not going to want to waste energy, as we have seen repeatedly before. So the case occurs, what if we need NADPH, but we do not need ribose 5-phosphate? The problem with this is that once we enter the pentose phosphate pathway by proceeding through glucose 6-phosphate dehydrogenase, this is irreversible. So we cannot reverse ourselves back to glucose 6-phosphate and place this back into glycolysis. So what we're going to do is enter this pathway. We must go all the way to ribose, and then the ribose is going to undergo some conversions between it and its partner xylose 5-phosphate. What I'd like you to recall simply is that we have enzymes called trans-aldolases as well as trans-ketolases. And this is a series of reactions that are able to take the 5-carbon ribose structure and through some carbon combinations, moving carbons around, we are able to reconstruct fructose 6-phosphate and glyceraldehyde 3-phosphate. These both, of course, can be placed back into glycolysis. The sole reason that the cell will do this, of course, is because if it does not need ribose, because it has plentiful DNA and RNA precursors, nucleic acids, nucleotides, then let's recapture this energy-rich molecule by going through some conversions and place it back into glycolysis so that we can continue extracting energy. The cost of this reaction, of course, is the loss of one carbon. So now we are losing a carbon going through this process. However, that energy is being harnessed in NADPH for reducing power. But most importantly, we are not losing the whole structure. Now, the complexity of the reactions between the transaldylases and transketalases is something that is unnecessary for our course to be bothered with. But keep in mind the complexity is that fructose is a 6-carbon molecule, glyceraldehyde 3-phosphate is a 3-carbon molecule, ribose is a 5-carbon molecule. So if you do the math, what we're looking at is that 3-ribose, which is 15 carbons, will be converted into 2-fructose 6-phosphate, which is a total of 12 carbons, plus 1-glyceraldehyde 3-phosphate, which is a 3-carbon, to balance the carbons in our chemistry.